Oh my gosh, thank you all so much for coming to seminar today. I'm so glad you guys have a good turnout. I am introducing Dr. Barnabas Daru. He got a bachelor's in science at the University of Jos in Nigeria, and then he went on to do his master's and PhD in botany at the University of Johannesburg in South Africa. And then he was a postdoctoral fellow at Harvard. And then I met him when he was an assistant professor after that at Texas A&M University, Corpus Christi. He's actually my first PI and taught me how to promote on and all these cool things. So we kind of go way back. And now he's an assistant professor at Stanford, and his lab mainly focuses on plant species distributions and how those species came to be distributed in the ways that they are. And ask questions about how that can better inform us about conserving plants and other species. So yeah, with that, I'll let Barnabas take it away and give us his talk.
Okay.
And while he's doing that, also, please join us for tea time directly after this in Pacific Day.
All right. Thank you so much. Thanks, Jordan, for that kind introduction. And I'm so excited to be here, and I've had wonderful times meeting with some students and faculty already, and I look forward to my meetings later on with more people. So yeah, thank you so much for having me here. And so today I'll be talking to you about my research, which I've entitled Exploring Contemporary Patterns of Plant Biogeography. And so as a way of introduction, I'll start with a brief introduction of what biogeography is, which is the study of the patterns and processes of life across the globe. And so this is based on the idea that biodiversity is not randomly distributed across the world, but it's instead clustered into communities such as the tropical rainforest or the chaparral of California or the woodland savannas of Southern Africa. And so my research is a step in this direction and seeks to understand the processes by which biodiversity is generated, is distributed, and maintained. And so along these lines, our lab seeks to address three basic kinds of questions. So for the first question, we seek to understand how do we determine which unique communities to conserve. And we also want to know where are species distributed today, and how or what are the underlying mechanisms that drive changing distributions in the Anthropocene. And so I've kind of outlined my talk along this line, starting with the way we seek to conserve unique communities, how to understand patterns of species distributions, and how to determine changing distributions in the Anthropocene. So for the first question, in terms of how do we determine what unique communities to conserve, so excuse me. Escape this. Sorry. No. Oh yeah, sorry. Anyway, yeah, so we asked the question, how do we determine which unique communities to conserve? And so here, I use a case study of the reorganization of seagrass communities in a changing climate. And this is work that was done in collaboration with my former graduate student, Brianna, who now works as a research scientist in Clearwater Research Institute in Florida. And so we focus on seagrasses. Seagrasses are not grasses, but they are monocots, and they belong to the order Alismatales. there are 7 families and 72 species distributed along the coastlines of the world. And these are some examples of seagrass meadows that you can find, like the eelgrass is very common along most coastlines of North America, as well as Cymodosia rotundata. And more recently, the genome of the eelgrass, Zostera marina, was published in Nature in 2016. In addition, seagrasses are also very important in the ecosystem, in the marine realm, because they are primary food for marine herbivores like the endangered sea turtle. They are also important nursery grounds for most fish and invertebrates. And so to understand how seagrasses are likely to respond to global change, we ask the following questions. So to what extent will the alpha diversity of seagrasses change under current and future climate scenarios? Will there be evidence of changes in seagrass phylogenetic beta diversity as well as species beta diversity under future climate scenarios? And how effective will the current network of marine protected areas be in terms of safeguarding the future of seagrasses under climate change? And so to address this, we use species distribution models, which is simply the use of species occurrences and maps of the environment, applying a statistical model in the form of machine learning models, for example, using maxn, and that allows us to generate a map of probability of a species being present in an area. And using that information, we can predict current distributions as well as project future distributions. So we apply this framework for seagrasses, and so we quantify changes in alpha diversity. Here, we model the distributions of species in the future, and we contrasted that with species predictions in the current time to have an estimate of changes in alpha diversity. We repeated the same process for changes in beta diversity by taking the difference between current species composition and future species composition. And then we quantify coverage by the current network of marine protected area networks. So when we did that, we found some really exciting results. So for the first one, here I show map of changes in species geographic ranges. And here, this under different scenarios of climate change, spanning from a best case scenario of greenhouse gas emission to a worst case scenario of CP 8.5. And so here the colors corresponds to predicted changes in species geographic range sizes, with the warmer colors corresponding to percentage range size loss, and cooler colors corresponds to range gains. And so we map this for the year 2050, and we contrasted that also to the end of century scenario in 2100. And so when we did that for all seagrass species, we found that there will be widespread range reductions in seagrass ranges varying from about 3 to 6%. So to understand if these patterns of change in species range sizes is phylogenetically patterned, we also map these predicted range changes onto the phylogenetic tree of all the seagrass species for both the mid-century scenarios in 2050 and end of century scenario in 2100. Again, the colors, warmer colors correspond to losses and cooler colors corresponds to range gains. And so we wanted to know whether closely related species tend to share similar changes in their ranges. And we found that there will be weak phylogenetic signal in range change, suggesting that other non-phylogenetic factors like the environment and anthropogenic change may be driving range changes in seagrasses. So we wanted to then understand the distributions of these changes in geographic space. And so we first mapped changes in alpha diversity as measured by species richness and phylogenetic diversity. What this means is that we first map changes in species richness in the current time. So here, cooler colors correspond to areas of predicted high richness and warmer colors, lower richness in the current time. We contrasted that with future and predicted range species richness in the year 2050. And then we took the differences between the current and future to have a difference map where cooler colors correspond to richness gains and warmer colors richness losses in the year 2050. And then we projected that also to the end of century scenario in 2100, and we took the difference as well. And so we also repeated the same for changes in phylogenetic diversity. So phylogenetic diversity simply quantifies the length of branches from the tip to the phylogenetic tree, to the root of the phylogenetic tree to encapsulate the shared relatedness of organisms. And so we mapped this similar to that of richness, and we found no significant changes in richness and phylogenetic diversity, except in tropical Indo-Pacific and the central Indo-Pacific as indicated in the map here in the red circles, where losses of alpha diversity will be pronounced. And so we then wanted to know if these changes also impact irreplaceability as measured by endemism. And so we mapped facets of species-weighted endemism and phylogenetic endemism. And these metrics really just quantify the degree of range restrictedness of species. And we contrasted that for current scenarios and future scenarios in 2050, as well as in end of century scenarios in 2100. And we took the difference as well. And so we found that there will be projected increases in weighted and phylogenetic endemism, confirming the range reductions that we found previously. However, though, the tropical Eastern Pacific will experience losses in endemism that will lead to invasion, meaning species in those regions are likely to experience projected range expansions. And so to then identify areas of richness that can, aspects of biodiversity change in seagrasses that can be decoupled from richness and phylogenetic diversity, we map beta diversity of seagrasses, and this is based on the differences in species composition among sites for current and future scenarios using species level beta diversity. We also contrasted that with phylogenetic level beta diversity, and we found that, again, there will be projected increases or gains in species and phylogenetic beta diversity across most regions that will cause seagrass communities to become differentiated, as illustrated in this chart and also the other chart in the end of century scenario, except in the Eastern Indo-Pacific and temperate Southern Africa as highlighted here, where beta diversity will decrease, that can lead to the homogenization of seagrass communities in the future, meaning seagrasses communities in these regions are likely to become more similar than expected randomly. And so we then wanted to know how well will each of these facets of diversity be protected by the current network of marine protected area networks. And so we overlaid the hotspots, that's areas that capture high concentrations of each facet of diversity, including the species richness, phylogenetic diversity, all the way to phylogenetic beta diversity across the current network of marine protected area networks. So here, the warmer colors correspond to tendency toward no protection, whereas cooler colors indicates high protection. And so this is protection levels for seagrass species richness hotspots for all the different scenarios, from a best case scenario in 2.6 to a worst case scenario of greenhouse gas emission. And so we mapped this for current scenario, mid-century scenario, as well as end of century scenarios in 2100 for species richness. And we did that for all the other facets of diversity, from species richness at the very top all the way to phylogenetic beta diversity at the bottom, and for mid-century scenario to the left and end of century scenario to the right. And regardless of the metric, we found that majority of the hotspots will not be protected within the percentage, will not be protected by the current network of marine protected areas, with the percentage of unprotection projected to increase of. So going back to the initial question in terms of how do we determine which unique communities to conserve? So we show that there will be projected reductions in seagrass species ranges that will lead to increases in areas of endemism. However, such changes will also result in divergent shifts in beta diversity that can lead some areas to become very differentiated, as well as other regions become more homogenized. And so the hotspots of shifts in alpha and beta diversity, however, will occur outside of the current network of marine protected area network. And this provides opportunities for prioritizing future conservation actions. So going back to the initial framework in terms of how research program seeks to understand patterns of species distributions. So we asked the question, where are species distributed today? And so for this, I'll talk to you about a recent project that we did, titled Mass Production of Unvouchered Records and How They Fail to Represent, global biodiversity patterns. And so this is work that was done in collaboration with Jordan Rodriguez, who graciously, gave me a very nice introduction this afternoon. And so at the time, she was an undergraduate intern in our lab. And so we wanted to know how these unvouchered records, the mass accumulation, these records, representing a knowledge of global biodiversity patterns. And so to do this, we know now that its data is increasingly becoming very crucial to address the ongoing biodiversity crisis. And some types of biodiversity data can come from things like museum collections. This is an example of collections in the Smithsonian, museum. And also data is also increasingly coming from other mobile applications like cell phones and other citizen science projects that capture information on organisms and increasingly, send them to repositories that all meet together at some point to form biodiversity data. Put another way, biodiversity data can be categorized into two: voucher records and observation records. So voucher records are simply sometimes referred to as primary biodiversity data, meaning they have links to a tangible physical material in a museum or a herbarium. On the other hand, and this diagram was generated by Jordan, and it is, observations are simply sightings of organisms. They do not necessarily have a link to a physical material in a museum or a herbarium. However, they are increasingly being used for addressing questions in ecology and evolutionary biology. These data sets, regardless of whether they are from vouchers or observations, also were not randomly collected, meaning they are prevalent with so many gaps and biases. And there have been efforts to understand the coverage gaps and biases in biodiversity data. For example, we recently were interested in addressing biases in museum collections of plants, herbarium specimens for the flora of Australia, South Africa, and New England in the US. And so here, areas in red corresponds to high coverage and blue low coverage. So you can see there's sort of an imbalance collecting in these different regions. So, but then we wanted to know whether these coverage gaps and biases, how different they are between vouchers and observation records. So to do that, we use a database of about 1.9 billion butterflies, birds, reptiles, and mammals, and we quantify coverage and biases of expected biodiversity patterns by voucher and observation records. But for the purpose of this talk, I'll just limit it to the coverage of vouchers and observations by plants only for now. And so we asked the question, to what extent is the coverage and biases of expected biodiversity is by voucher and observation records? And so here we define coverage as the number of records that are required to inventory species in terms of their richness or abundance along taxonomic, geographic, temporal, and functional trait dimensions. And we found some really exciting results that I'd share with you today. So first, when we map taxonomic coverage of lineages, so here, these are the lineages of plants corresponding to plant families. And so we have voucher records on the left and observation to the right. And so the scale here corresponds to the degree of coverage with darker colors, meaning that the records of vouchers or observations capture our expected knowledge of richness of species within a family, and lighter color corresponds to a scenario of no coverage at all. And so we found that observation records tend to show phylogenetically biased coverage and less congruence with expected patterns of plant richness more than voucher records. And so when we mapped this and contrasted it space, we found that there is severe sampling biases in biodiversity-rich regions like South America, Africa, and Southeast Asia, and so so there's more coverage by voucher records than observation records in these biodiversity-rich but infrequently sampled regions of the world. And so then we wanted to understand the degree of geographic coverage of species and regions. First, we quantify the density of sampling per species for voucher records and records indicated in gray. And we found that there is significant biases of geographic density of records under observation records than for voucher specimens. And so when we map this again across geographic space, we found that South America, Africa, and Southeast Asia tend to have fewer observation records compared to voucher specimens. And then we want to know how this plays out across a temporal scale by mapping temporal coverage. So here we wanted to know how the time interval between one collecting event to the next, for species is playing out for both vouchers and observation records indicated, voucher records indicated in blue and observations in gray. And we found that voucher records tend to show much better or higher temporal coverage for most species compared to observation records. And when we compare this across geographic space, we found that observation records tend to have larger temporal gaps, especially in mega diverse and infrequently collected regions like Africa, South America, and Southeast Asia. And most of these regions tend to have large collecting gaps, meaning there's a huge time span between when a specimen was collected to the next, providing opportunities for guiding collecting efforts in those. And so then we also explored how well the coverage of these records are in terms of the intrinsic life history traits by mapping different facets of functional trait diversity, including duration, habit, maximum height, seed size, and even extinction risk for both vouchers indicated in blue and observation records indicated in gray. And so the bars correspond to the density of records, numbers of specimens per species. And so we found that observations have more records per species, but uneven coverage of functional traits. Whereas on the other hand, voucher records tend to have more evenly distributed coverage of intrinsic life history, despite being not having very few records per species, but the evenness is much more covered as opposed to observation records. So we then we asked the question, what might be driving these factors? What sort of factors underlie the patterns that we're seeing? So we explored the potential of various ecological and socio-economic variables in driving coverage in terms of geographic, taxonomic, and temporal coverage for across sites. And we use various factors like elevation, wilderness correspond to how remote an area is, and limited ground transportation, distance from airports, or the security of regions, as well as even the research funding of different countries or taxonomic, geographic, temporal coverage. And so we found that coverage patterns are driven largely by how accessible and secure a sampling site is, and in turn reflect highly human modified areas. So here the dots correspond to just the estimated coefficients from a spatial error model, and the error bars corresponds to 95% confidence intervals for both voucher and observation records. So going back to the initial question that we asked, where are species distributed today? So when we contrasted coverage by vouchers and observation records, we found that the nature and severity of coverage of expected biodiversity patterns differ between voucher and observation records. And such differences in coverage by voucher and observation records have implications on how these records are used for research in ecology and evolutionary biology. However, though, these data sets, both vouchers and observation records, are also complementary and vital resources for assessing how anthropogenic drivers affect biodiversity. So going back to our initial framework in terms of how our research seeks to understand changing distributions in the Anthropocene. So we asked the question, what are the mechanisms that drive changing distributions in the Anthropocene? So here I use a case study titled The Homogenization of Plant Community, the Anthropocene. And this is work that was done in cooperation with different labs and groups, for which I'm really thankful for, and being able to work with these amazing individuals. And so I'll take you a little bit on the history of us, the last 540 million years ago. And so I just highlighted the last 23 indicated here are the major, in asterisk at the bottom, are the major mass extinction events. And we have at the top the last 23 million years ago, including Miocene to the left, and the Pliocene, the Pleistocene, the Holocene, and then there's a new epoch that is beginning, is sort of under question, and indicated in the icons are major fauna that radiated during each of the epochs. And so this last epoch under question is beginning to somehow be called the Anthropocene, largely because it's an epoch that is driven by profound human impact on the environment. And during this time, we are witnessing high human population growth. And at the same time, the climate is changing rapidly. And species are also going extinct at an unprecedented rate. And at the same time, species are also being dispersed outside of their native ranges to become naturalized in other regions. And so for purposes of this presentation, I'll focus on the last two processes, species extirpations and naturalizations, and how these two can combine together to form what's referred to as biotic homogenization. And so this is simply the reduction of trait and phylogenetic differences in species that are shared between regions. And these can diminish the regional distinctiveness of regions and can have negative impacts on key ecosystem services. So to understand how this concept of biotic homogenization is playing out in plant communities, we first assemble a database, which we call green maps of species distributions. And so we did this for over 200,000 plant species across the globe. And so this is just a map that shows the richness, species richness of species across 100 by 100 kilometer grid cells, where areas in red correspond to high richness and blue low richness. And it matches what is expected, the expected knowledge of plant distributions across the globe. And on the right there is the recent phylogenetic tree of vascular plants across the globe. And so we then quantify changes in alpha diversity in the Anthropocene, and taking the difference with that of the Holocene. Put another way, alpha diversity correspond to species composition after any form of naturalizations and recent extirpations minus species composition before naturalizations and extirpations. And just to remind you again, our definition of Anthropocene and Holocene correspond to these two time epochs, the Holocene to the left and the Anthropocene to the right. And so we did the same for changes in beta diversity by taking the difference in species composition between the Anthropocene and the Holocene. And then we quantify the asymmetric exchange of non-native phylogenetic diversity across regions. So when we did first, we map changes in species alpha diversity in the Holocene. That's before any form of recent introductions or extirpations using species richness and phylogenetic at the bottom. Again, areas in red corresponds to high richness or high phylogenetic diversity, and blue, low richness or low phylogenetic diversity in the Holocene. We contrasted with changes in alpha diversity in the Anthropocene after accounting for recent introductions and extirpations for both species and phylogenetic diversity. And we took the difference. And so here, blue areas correspond to losses in alpha diversity, and warmer colors corresponds to gains. And so we found widespread patterns of reductions in alpha diversity in the Anthropocene. And so then we wanted to know, how is this changes in plant communities sort of decoupled from patterns of richness and phylogenetic diversity? And to do that, we map facets of beta diversity. However, though, for beta diversity, because it decays, beta diversity decays with geographic distance. So we quantify beta diversity within what's referred to as the current networks of botanical biomes, sort of corresponding to country borders, but capturing sort of differences in species composition across plant communities across the globe. And so we map beta diversity based on the Holocene species composition, that's before any form of introductions or naturalizations, and species composition of beta diversity and phylogenetic beta diversity in the Anthropocene after accounting for extinctions and introductions. And we took the difference, and again, the lighter or warmer colors corresponds to gains in beta diversity, and cooler colors correspond to losses in beta diversity. And here we found widespread patterns of loss in beta diversity that can lead to homogenization of plant communities. And some of the places that are projected to experience the greatest shifts in beta diversity, parts of North America, as well as Eastern Australia, as places with projected losses in beta diversity that can lead to plant communities. We then we explored this biotic change even across different scenarios of extinctions and introductions, spanning from a scenario of no extinctions to a scenario when we assume that currently threatened species are to go extinct, and we found that changes in plant communities are driven largely by non-native naturalizations rather than by native extirpations. And even when we assume that currently, a worst-case scenario where currently threatened species are assumed to go extinct, we found that non-native naturalizations still remain by far the strongest contributors to biotic reorganization in the Anthropocene. So then, then we wanted to know how the exchange of these non-native species play out across continents. So first, for example, which species, we quantify the directionality of species that are native to a region and being exported to other parts of the world. For example, native species in North America and being introduced and naturalized in other parts of the world, or species that are native to Africa and being introduced to other parts of the world. And we did that for all the continents. And so the thickness of the lines corresponds to the proportion of species that are being exported across different regions. And the same is true for the directions of the arrows as well. And so we mapped the exchange of non-native species across continents, and we did the same for the exchange of phylogenetic branch lengths across continents as well. And even we corrected for phylogenetic branch lengths with species richness in this last chart to account for differences in species richness. We found that Asia and North America are the major sources of non-native species, but the species that they export tend to be phylogenetically close to the recipient floras. Australia and Europe, on the other hand, contribute very few species to the global pool of non-native species, but they represent a disproportionate amount of phylogenetic diversity. So going back to the initial question, in terms of what are the mechanisms that drive changing distributions in the Anthropocene, so we've showed that regardless of the extinction scenario, the strongest contributors to biotic homogenization results from non-native naturalizations. And Asia and North America are the major sources of non-native species, but the species that they export tend to be phylogenetically close to the recipient floras. And so this kind of results demonstrate the profound influence that humans exert on regional biotas beyond changes in species richness. So putting all this together, so far I've shown how our research helps to identify which unique communities to conserve and how there is differences in coverage between vouchers and observation rates, and how we can understand changing distributions in the Anthropocene. And so ultimately, our research program seeks to contribute to elucidating the mechanisms by which biodiversity is generated, is distributed, and maintained in the Anthropocene. So for some of the things that we are in terms of future directions, so we have three things that we're currently very interested in: looking at plant adaptation to life in the sea. We also are interested in developing computational tools for addressing questions in macroecology and biogeography, and we want to understand macroecology in the Anthropocene, that's in the modern century of profound human impacts. And so for this first question, for understanding plant adaptation in the marine environment, so we are using seagrasses as our model system to understand how the evolutionary transition from land back to sea played out in seagrasses, which happened around 140 million years ago. And to do this, we are looking to explore the genes that are involved in the formation of cell wall polysaccharides in seagrasses. And we also want to explore the microbial symbionts that confer life, or that help to support seagrass environment. And so for the second work, we develop tools for macroecology and biogeography, largely because some of the tools, the existing tools, were initially developed for addressing questions at scales that represent our community ecology, and may not scale out for addressing questions across larger temporal and geographic scales. And so we are developing a tool called green maps to help address the shortfalls in the distributed, as indicated in the previous project on biases and coverage gaps. And so to do this, we want to ultimately use this data to quantify changes in species range sizes for plants through time. And that can also help us to address questions like the tempo and mode of plant diversifications across mountain ranges. And we hope to ultimately develop a mobile app that can help steer users to places that have not been collected before, as opposed to randomly just collected across the landscape that can easily lead to biases and skewed coverage. And ultimately, we are also planning a workshop. In fact, there's a workshop coming up in June of this year. So please be on the lookout for those who might be interested in learning patterns of biogeography in a workshop setting. So yeah, and then for the third objective to quantify macroecology in the Anthropocene, we simply want to understand what mechanisms drive changing or the unbalanced exchange of non-native species. So based on the idea that species exchange across continents are so imbalanced, so we've wanted to build from the homogenization project to explore the biogeography of non-native plant exchange between the US and South Africa. And so why these two regions is simply because the biomes in the region tend to show a bit of conservatism. For example, the Mediterranean California region tends to mirror the most of the Cape region in South Africa. The temperate desert mirrors Namakaru, as well as the great Van of South Africa and so on. So the question is, as a species, for example, that originates from the fynbos of South Africa and is introduced to North America, its success likely tied to the kind of biome that is associated with, or is it random? And vice versa. And so we want to explore the mechanisms that drive this sort of exchange of non-native species. And to do this, we want to look at the biome conservatism of species invasion, and looking at also the genetic changes that may play out from the native range to the introduced range. And we also plan on doing some workshops across the different regions, spatial targeting minoritized groups in these two regions that we are looking to explore. So put together, all this research and projects that we did would not have been possible without the help, and support of all these amazing people that have had the chance to pass through the lab. And so these are some members of the lab, both past and current members of the lab. And the work also has been really supported by various institutions like Stanford and the NSF, as well as from the amazing group of those I've had the chance to work with as well. And thank you for listening. Yeah.
So, really interesting stuff. I have two questions. So you've shown that human mediated dispersal or non-native naturalization, as you call it,
Right.
species, contributes to homogenization, so that's a better diversity. But can you clarify whether non-native naturalization contributes negatively or positively as a diversity? Because the effects shown included extinction as well as non-natural
Right.
But if we're not focused upon native naturalization, does that contribute positively or negatively?
Yeah. Oh, yeah. So you're asking about if the changes in species composition as reflected in alpha diversity, whether it impacts beta diversity negatively. Is that?
Oh, whether human mediated dispersal contributes positively or negatively to local richness.
Right. Yeah. So that's a really great question. And so to address that, one of the things that we are beginning to look at is these changes in plant composition may depend on the scale at which it plays out. So, and there's really an ongoing debate as to whether or not species composition is, whether it's actually increasing or it's decreasing. And so the question, may depend on the scale at which you're looking at. So as at certain regions, because species invasions vary across landscapes, so some places are likely to have more invasion than others. And at that, scale, it may look like alpha diversity is increasing. But if one scales out by looking at differences in species or phylogenetic composition, then it may show, how to decouple that from patterns of richness. And so we thought looking at beta diversity may help us to see, yes, despite some places may experience increases, but that increase may not necessarily result in increased distinctiveness of regions, but it may lead to homogenization or the loss of beta diversity, because species, are found everywhere. And so there's sort of loss of turnover across regions, as opposed to what will help to increase the differentiation of regions. Right. Yeah, that's a great question. So you're asking about the sources of data to characterize changes between the Holocene and the Anthropocene. Yeah, so our definition of the Holocene and Anthropocene is very loosely defined, and it's not necessarily tied to a time point per se. So we define species composition in the Holocene, that's before any form of human impact or profound impact on the environment, by simply quantifying beta diversity or change without accounting for any recent introductions or extirpations. And then species composition in the Anthropocene is when we incorporated information on the species introductions, naturalizations, and also taking away species that either threatened or those that have been recently documented as gone extinct, and to see how changes play out across plant communities. Great question. Right.
For example, human agriculture, such as taro that kind of like spread out 5,000 years ago, is that considered Holocene or Anthropocene?
Yeah, absolutely. Yeah, those are really great questions. So asking about what. Yeah, that's really great question. Again, asking whether introductions that play out, say, about 1,000 years or so ago during the hunter-gatherer times can be, likened to introduction. So that's also another ongoing debate in the invasion community as to really what constitutes a species or an invasive species. So, for example, the flora of most of Europe has long, been naturalized to some extent that it can be considered native to the region. And so our definition of species invasion in this context may loosely correspond to the start of the Industrial Revolution. That's when humans actively started moving things across continents, sort of like through the Columbian exchange, So yeah, so that's a really great question. And it's also one that we want to know at what extent, how scale dependent is invasion, in terms of even the time when they were introduced and even the geographic scope. So can a species that is native to the New England of North America and introduced to the California region, can it be considered invasive, even within the same continent? So those are great questions that we think about a lot. And thank you for that question. Yeah.
So you may have done looking at asymmetry given the biogenetic input
right.
That analysis is, but probably that analysis accounts for phylogenetic diversity that's present in continent of the beginning. For instance, Australia, world land quite clearly.
Right.
It might represent pretty disparate on phylogenetically, even before I started moving. So do you know costs account for what is the inherent phylogenetic diversity, and then ask, okay, given that it's industry, and I moved it from Australia, and it has X% phylogenetic diversity.
Right.
Does it account for what the phylogenetic diversity is like in that continent is? The reason I ask this,
Right.
is because it's notable to me that a lot of the reproductions aren't changing phylogenetic.
Right.
It's almost like the environment selecting which species potentially to take off.
Right.
So you how that phylogenetic diversity is unequal across the globe.
Right. Yeah, absolutely. You know, looking, asking about how equal or unequal is the exchange of phylogenetic diversity across regions, bearing in mind the evolutionary history of the region. That's a really great question, and also one that is very key and central to our research program. So we ultimately want to know, you know, how phylogenetic information contributes to our knowledge of biodiversity beyond just species richness alone. And so we, for our study on the exchange of, you know, phylogenetic branch lengths across regions, species are not necessarily independent units, but they represent, they sort of have shared ancestry in terms of their phylogenetic history. And so, analyzing that helps us to, you know, like the question of whether species richness is increasing may, you know, help us to understand whether that increase in phylogenetic or in species richness actually translate into increases in phylogenetic diversity. Put another way, whether the species that are coming into a region are likely to represent closely related species, or, you know, in another way, is sort of testing this Darwinian hypothesis of enemy release hypothesis, or you know, whether as a species is introduced to an area, is its success tied to the fact that it has close relatives around or not? And so we wanted to test this at this global scale, and so that's the, you know, rationale behind that analysis. Yeah.
Happening for a long time, and that sort of differences between the temperate seagrasses and the more tropical ones.
Right.
Whether you saw a difference in the, I noticed, for example, that you could project at warm cooler
Right.
for the temperate class.
Right. Yeah, absolutely. That's a really great question. So asking about whether there's a difference in the projected changes in composition between the temperate seagrass, you know, biomes and tropical ones. And so, yes, in a previous study where we mapped the biomes of seagrasses in terms of their shared evolutionary history, we found that there's a latitudinal gradient in evolutionary distinctiveness of seagrass communities, with the temperate regions being more evolutionarily distinct than the tropical regions, suggesting that perhaps other historical factors like extinction may have played out in shaping the distributions of species in the temperate region, leading to the distinctiveness that we're seeing. So put another way, it's possible that species in the temperate regions may represent once very diverse groups that may have become lost, you know, through extinction processes in the past. And so the differences that we are seeing may also, you know, partly be explained by sort of the possible introductions or extinctions that may have played out for temperate regions, like glaciation events and things like that, shaping the patterns that we're seeing. But the fact that we also didn't see a link with phylogenetic relatedness may also reflect the profound impacts of human activities on, seagrass communities in these regions, indicating that impacts may be very unbalanced across temperate and tropical regions.
We have genera that have species that are found in very cold regions as well as very.
Right. So in terms, yeah, absolutely. That's really great question. So asking about how deep the diversity of seagrasses are, in temperate versus tropical region. So in terms of the center of diversity of seagrasses, it is concentrated in the Indo-Pacific region of, in the tropical region of the world. But then the temperate regions tend to have show sort of a disjunct distributions, and looking at it phylogenetically, it actually turns out that these disjunct populations are closely related, suggesting that they may, have been connected before, but, because of other mechanisms like dispersal or extinction, then, these species are now becoming very disjunct, but may have been connected before in the past. Right. 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 Yeah, absolutely. Great question. That's like the elephant in the room, right? For most species distribution modeling efforts, and how well the model is also has a function to do with the input data. And so whether we incorporated things like biotic interactions in our models, we would, but such data is not available. And that's also one of the reasons that, inspired us to quantify, coverage and in, functional traits, for example. And yeah, so yeah, if those such data set is available, we would really love to do that. And one way that, we are curious to explore going further is to look at things like microbial symbionts and how that the interactions between, seagrasses and their microbial symbionts are responding to global change. So whether or not there is a decoupling of the interaction that, of such either mutualistic relationships that are now becoming either pathogenic or not, and things like that. Those are some of the areas that we would love to explore. Yeah, really great question. Thank you.